In the dim light of a cold dawn, on the edge of a fishing dock in Nova Scotia, crates are quietly lifted from boats still slick with salt. Inside each box, nestled in darkness and foam, is a life waiting to cross an ocean, not as cargo, not as meat, but as a living organism suspended in a delicate pause. A lobster, armored in shell, yet biologically fragile. What follows isn't just shipping. It's a thermodynamic choreography where biology, chemistry, and logistics must agree every single hour. Lobsters are among the most challenging live cargoes in global cold chains. Their survival depends not only on refrigeration, but on oxygen supply, air pressure, stability, and real-time gas exchange. A lobster doesn't breathe like a human or travel like a fish in water. It survives through gills, in a humid environment just shy of saturation, where every thermal shift threatens cellular collapse. Technically, it's not unlike transporting cryogenic tissue samples for medical research, except the lobster must stay alive, not simply preserved. The containers used for lobster freight are not ordinary boxes. They're biomechanical capsules, insulated chambers designed to absorb shocks, manage moisture gradients, and prevent gas saturation. In many ways, they resemble the core sample carriers used in mining, where preserving the chemical integrity of the sample is more critical than its physical weight. These lobster crates must do the same, but for something alive. They are built with impact-dampening walls, foam inserts to minimize gill abrasion, and lining materials that emulate the moisture retention systems used in post-harvest fruit transport. At the heart of their function is a principle known as temperature inertia, the system's ability to resist internal thermal change despite fluctuations in external conditions. Borrowed from agricultural thermal modeling, this principle was once used to calculate how mangoes resist heat loss during international freight. Now, it informs the way lobster boxes are layered with thermal buffers and humidity membranes. The goal isn't simply to cool, but to delay the rate of change, because even a 2-degree shift outside the safe zone can trigger stress, hypoxia, and eventual death. A graph appears. On one axis, pressure and oxygen levels. On the other, temperature and time. The optimal survival window, between 2 and 4 degrees Celsius. Relative humidity near 95%. Carbon dioxide levels held just low enough to prevent acidosis, but not so high as to trigger anaerobic stress. Every variable is monitored, predicted, and adjusted, because this isn't just food in transit. It's life, encased, engineered, and airborne. To move a lobster from ocean to dinner plate across continents is to operate on the edge of biological feasibility. And it all begins here, with a box that doesn't just contain a crustacean, but sustains a marine environment in miniature. What seems simple is, in fact, a mission of precision. One box, one load, one biological system in motion. Inside a sealed cargo hold at 10,000 meters, a subtle but lethal process can unfold. Unnoticed by sensors, undetected by crew, and unstoppable once triggered. Lobsters, stacked in ventilated polystyrene crates, appear calm, but under the foam lids, invisible chemistry builds. Minute by minute, the carbon dioxide they exhale accumulates, and unless it's properly vented or chemically neutralized, it turns into a silent suffocation chamber. In most transport boxes, ventilation relies on passive airflow, small perforations meant to exchange gases as pressure fluctuates. But this system, designed for fruits or vegetables, doesn't account for respiration in live animals. Lobsters, trapped in under-ventilated crates, begin rebreathing the same air within 8 hours, especially if there's a customs delay or a pause on the tarmac. CO2 concentrations can reach thresholds that disrupt their acid-base balance. The result is stress-induced hyperactivity, followed by systemic collapse. Not from cold, not from shock, but from the slow, tightening grip of gas toxicity. To prevent this, some exporters have turned to unlikely inspiration: mushroom farms. In the world of controlled fungal cultivation, CO2 levels are tightly regulated using biofilters. adaptive layers of activated carbon and bicarbonate matrices that absorb and balance respiratory gases. These same principles now guide the next generation of lobster boxes. Inside their walls, thin cartridges capture carbon dioxide while maintaining stable humidity. It's not just passive airflow anymore, it's targeted biochemical management. And yet, increasing airflow alone isn't the answer. A Canadian study tracking lobster mortality during static air freight simulations found that higher airflow, when unregulated, often caused thermal instability. Warm air entered faster, cold escaped. The result? More dead lobsters, not fewer. In one configuration, mortality rose by 18%, simply due to overventilation. What kept them alive wasn't more air. It was regulated exchange, just enough to remove CO2 without disrupting thermal inertia. In 2019, a shipment from Nova Scotia to Shanghai highlighted the stakes. A 1.2-ton load of live lobsters suffered a 27% loss within hours of arrival. The cause? CO2 buildup during an extended customs delay. Boxes had been sealed tight to preserve temperature, but without gas control, they became traps. That incident is now used in training modules across the cold chain industry, not as an anomaly, but as a lesson in environmental precision. Because what keeps a lobster alive mid-flight isn't just oxygen. It's a constant, hour-by-hour balance between temperature, gas composition, and metabolic demand. It's a system that breathes, responds, and adapts, even as it crosses continents at the speed of a jet engine. To the untrained eye, a lobster in a foam crate might seem no more complex than a chilled fruit in transit. Both are perishable. Both require stable temperatures. But beneath the shell, a lobster carries an entire marine chemistry, one that doesn't stop functioning the moment it's boxed. Unlike an apple, which remains chemically inert, a lobster continues to breathe, excrete, and interact with its confined environment. That metabolic continuity turns logistics into a biological balancing act. Under stress, lobsters emit ammonia and lactate, byproducts of survival mechanisms triggered during hypoxia or handling. In an enclosed, semi-humid box, these compounds quickly alter water chemistry. pH drops. Ionic imbalances develop. And unlike water shipments, where dilution mitigates these changes, dry air boxes have no buffer. The lobster ends up in a chemically unstable microclimate, a closed-loop environment where its own biology begins to work against it. To counter this, researchers have developed nano-coated mineral wool liners designed to mimic the absorptive structure of sea sponges. These liners don't just soak up moisture, they actively capture excreted compounds and help stabilize ionic concentration. Built from microporous fibers, they delay chemical shifts by binding ammonia molecules and maintaining a soft humidity envelope around the animal's gills. In effect, they transform a passive box into a reactive habitat. The results are measurable. In controlled laboratory trials, lobsters transported in crates lined with ionic stabilizers survived up to 42 hours without water, compared to 24 to 28 hours in standard polystyrene containers. That difference isn't marginal. It defines whether a shipment arrives viable or becomes a total loss. In high-value markets like Tokyo or Dubai, those extra hours determine whether a single crate generates revenue or triggers insurance claims. One distributor in Japan took this insight further. Over a 6-month period, they retrofitted their cold chain system with multi-layer alginate membranes, biopolymer films capable of moisture regulation and ionic exchange. Before the change, their live arrival rates averaged 78%. After the retrofit, that number rose to 95%. Significantly reducing spoilage costs and positioning the brand as a premium supplier of live North Atlantic lobster. This isn't seafood transport, it's mobile marine engineering. Every crate carries not just a lobster, but a living chemical ecosystem, volatile, active, and autonomous. A lobster box must act like a miniature ocean basin, engineered to buffer the fluctuations of air travel, warehouse wait times, and temperature drift. Because while the apple waits passively to be consumed, the lobster is still fighting to stay alive. And in that difference lies the true complexity of modern, live cargo logistics. At first glance, transporting a lobster across continents may seem like a matter of keeping it cold, cushioned, and contained. But when examined through the lens of modern logistics and biological engineering, it becomes clear that the lobster is no longer just a product. It's a mobile, chemically active life form, a marine organism whose survival depends on the precision of multiple systems operating in perfect synchrony, from the instant it leaves seawater to the moment it arrives at a customer's hands, often thousands of kilometers away. This shift, from viewing the lobster as perishable to recognizing it as biologically autonomous in transit, has redefined how we approach the cold chain. Traditional logistics treats the cargo as passive. It assumes the object doesn't change, doesn't respond, and only needs protection from external threats. But lobsters are different. They metabolize. They react. Their physiology shifts under stress, and that stress can be amplified or mitigated by the design of the environment around them. What emerges is the concept of a biologically integrated transport model, where the container isn't just holding the animal, but actively coexisting with its living processes. To meet this challenge, engineers and marine biologists are turning to cross-disciplinary innovation. In Norway, the Live Shell Initiative has developed transport containers for live Arctic crab that go far beyond insulation and airflow. These are self-regulating ecosystems, bioreactive units equipped with thermal shock correction systems and neural activity monitors. When the internal system detects a spike in crustacean movement, often an early sign of thermal or oxygen stress, it responds by adjusting internal temperatures or initiating microcirculatory ventilation. It's a feedback loop between biology and machine. The container doesn't just carry life, it protects it in real time. But the evolution of live seafood transport doesn't stop with Arctic innovations. In Asia and Europe, new systems are borrowing from the most unexpected sectors, including poultry farming and precision agriculture. Consider the egg transport cradle. Designed to safeguard fragile embryos during long-distance shipping, these cradles use suspension layers and vibration-damping geometry to absorb micro-movements that would otherwise fracture the shell or disrupt development. That same principle is now being adapted to lobster trays. Multi-tiered platforms with anti-vibration zones reduce mechanical stress during loading, unloading, and turbulence. It's a lesson transferred from land to sea, and now to air. What unites these approaches is a growing awareness. The lobster doesn't adapt to logistics. Logistics must adapt to the lobster. This means thinking in systems, not in units. Each stage of the journey, from the dock to the pallet, from the warehouse to the customs checkpoint, is no longer a segment of transit. It is a micro-environment with its own risks and variables. Time delays change gas composition. Temperature gradients affect gill hydration. Humidity loss impacts the buffer systems within the box. All of it compounds. All of it matters. This systemic perspective invites us to ask a deeper question. Is the lobster truly in transit? Or is it inside a modular marine chamber, temporarily divorced from its habitat, but still dependent on oceanic principles to survive? When engineers design these systems, they aren't just building coolers. They're replicating micro-oceans. They are managing oxygen diffusion, carbon dioxide saturation, ammonia neutralization, and ionic concentration. Each box is a fragile balance of thermodynamics, fluid mechanics, and biofeedback. The result is something profound. In this high-stakes choreography, every crate becomes an extension of the sea, engineered to keep life viable, not by halting biology, but by integrating it. And that changes everything. No longer is cold chain logistics about keeping products fresh. It's about maintaining homeostasis. It's about building containers that support respiration, delay metabolic fatigue, and extend viability with minimal biological cost. The lobster becomes a co-pilot of its own survival, supported by algorithms, sensors, and human decision-making. But this approach also redefines responsibility. It requires us to consider not just the mechanical resilience of a shipment, but the biological dignity of the species in transit. The more we understand their internal chemistry, their tolerance thresholds, and their survival curves, the more refined and respectful our logistics systems become. This isn't just transportation, it's stewardship, applied through engineering. Looking ahead, the frontier of live marine cargo may no longer be about duration or distance. It may revolve around adaptation, the ability of the system to adjust moment by moment to what the organism needs. It's possible that the containers of the future won't just maintain optimal conditions. They'll learn, respond, and evolve during the journey. Biology and machine merged in motion. So the next time a lobster crosses an ocean, wrapped in a foam crate and a layer of condensation, it's not simply a luxury product in a chilled box. It's part of a global choreography, a modular life form gliding through layers of engineering, calculation, and care. Quiet, invisible, and astonishing in its complexity. This is the living chain behind what the world eats. And it begins with a single organism, in a box, designed not to carry it, but to keep it alive, exactly as the ocean would.